All right, ladies and gentlemen, welcome back. So we're going to start digging into the actual internal structures of our cells. Before we do that, there's just a reminder here that when we look at cells and their classification, initially they get divided into two categories. Do they have organelles or do they not? And so that's your prokaryotic versus your eukaryotic, right? Prokaryotic cells are considerably smaller. They've got a lot less internal structure. They don't have those membrane-bound organelles. They don't have a nucleus, as opposed to your eukaryotic cells, which tend to be a little bit larger because we can increase that internal surface area by having all of those internal membrane-bound organelles. And then the eukaryotic cells get classified even further into plant versus animal, depending on what sort of internal structures that they have. And then we can divide even further down the line into specific types of animal versus plant cells. But for now, we'll just kind of stay on the surface here. So just a reminder, going back to your prokaryotic cells, right? These are your bacteria or your archaea, depending on their cell membrane structure and their evolutionary history. These guys lack those membrane-bound organelles. The only sort of organelle, quote unquote, that they do have are ribosomes, but ribosomes don't have a membrane around them, so again, they lack membrane-bound organelles overall, but your ribosomes are your site for protein synthesis. So every cell is going to have those ribosomes because they need a way to take their genetic information and convert it into functional proteins, and that's where ribosomes come in. Again, the prokaryotes don't have a true nucleus. They have sort of that nucleoid region in the center where we have the main bacterial chromosome hanging out. There's no membrane separating it from the rest of its cytoplasm. So when we sort of compare the two, right, your prokaryotic cells are smaller, they're much more primitive. Go back to the evolution, we believe these guys were the ones who evolved first. They've got that circular DNA located in that nucleoid region, but there's no true nucleus structure. There's no membrane separating it from its cytoplasm. Pretty few specialized parts. They'll have those ribosomes. They may have some infoldings of membrane to do things like photosynthesis or respiration, but there's no true chloroplast or mitochondria. And so we're looking at those bacteria or those archaea. Your eukaryotic cells, though, larger, more advanced, they can be bigger as a result of having all of those specialized cell parts, those membrane-bound organelles, because we can increase the internal surface area to make up for the volume and the lack of surface area that we would see directly on the outside. So we increase their efficiency by having all of those internal membrane-bound organelles. And then we're talking plants, animal, fungi, protists, those sorts of things. So when we look at prokaryote versus eukaryote, there are several features that regardless of what kind of cell we have, they all have these structures in common. First off, they all contain cytosol, or what you probably learned as cytoplasm in the past. Cytosol is specifically the liquid portion of that cytoplasm, and most of it's water, 90+% of it is water. Cytoplasm is sort of more just the area that we're talking about. They all contain chromosomes. They all have to have genetic information in some form. Generally speaking, we're talking DNA. We've got those genes, that set of instructions to tell the cell how to function. They will all have ribosomes as a way to have a place to take that genetic information and convert it into proteins, because proteins really are going to be your molecules of both structure and function in cells, and they all have to be bound by at least a plasma membrane. We'll certainly see that prokaryotes and even some eukaryotes have additional layers beyond that plasma membrane, but every cell has at least that. So speaking of that plasma membrane, let's just kind of look at some structure briefly here. When we get to chapter 7, we'll dive into this a little deeper. But remember, your plasma membrane functions as a selective barrier. We'll talk about it as being selectively permeable. It allows certain things in while keeping certain things out, and vice versa, certain things in, certain things out, and only certain things can pass across that membrane. So it forms a barrier between the cell's internal environment and its external environment. When we go back to evolution, that was a really big jump because now I could much more efficiently allow metabolic processes to occur by keeping them contained, by keeping molecules closer together. So that plasma membrane really forms that barrier. It does have to be large enough compared to the volume, right? Going back to that surface area to volume idea to allow sufficient passage of nutrients and wastes. So the cell really is limited by that plasma membrane surface area in terms of how much volume we can deal with. It is a phospholipid bilayer. So if you remember your phospholipid structure, right, we have those hydrophilic heads here, those little gray things that we're seeing here. Those are sort of the polar heads, hydrophilic heads. That's what the phosphate group is connected. So that phosphate has a charge, which is why it will interact with the water on either side of the membrane, both external and internal in the cytoplasm. And then you have that hydrophobic region in the center here, those fatty acid tails that don't like that water at all. And so they're going to bury themselves in the inside of that membrane to stay away from the water on either side. Keep in mind that that phospholipid is an amphipathic molecule, right? So it has a hydrophobic and a hydrophilic region. And it really is going to be those fatty acid tails that drive what kind of molecules are easily able to pass that membrane and what isn't. That does then complicate systems a little bit because there will be things like water that need to pass through that membrane but can't interact with those fatty acid tails. And so we've got to have membrane proteins then that are there. And one of the functions of those membrane proteins is to be transport proteins to allow certain things that wouldn't normally be able to pass just straight through a complete lipid bilayer, water being an example, to allow that water to get in. It's got to pass through proteins that kind of shield it from those fatty acid tails and allow it to have a hydrophilic region all the way through. Other membrane proteins also act as receptors, so as a way for cells to communicate with each other. It's all molecular, so they have to have ways to receive those signals and then cause changes inside the cell. So that's the idea of the receptors. Enzymes, same thing, same idea. Being able to catalyze chemical reactions, some of which may be in communication pieces, others for other reasons. Most of them will also have these carbohydrates sitting on the outside. These are kind of tags for ways for cells to recognize what kind of cell this is, where it is located, that kind of thing as well. So we'll get into some more detail in terms of that plasma membrane structure and what it does when we get to chapter 7, but these are some of the big highlights. Nice diagrams here just to kind of study. This is your animal cell, I should say. You've got all of the major structures here listed for you, identified for you, as well as some basics on their structure, or excuse me, on their function. So definitely something to take a look at. Also gives you a nice little quick brief what's in animal cells but not plant cells as a way to sort of tell the difference from one to the other. Keep in mind, while we're on this topic here, these are models of cells. We pretty much shove everything that could potentially exist in a eukaryotic cell, in this case an animal cell, into one image. This is by no means what a real cell would look like. Not every animal cell has this shape, let alone has all of these organelles in any sort of concentration that this picture shows. So remember, these are models, not accurate representations of real cells. And same thing would go for that plant cell here as well. So nice things to study from, nice things to provide you a way to understand the content, but again, not a realistic diagram of what a plant cell would actually look like. So a couple of major pieces, if we're talking eukaryotic cells, across the board, they all have to have genetic material. So that means they've got a nucleus in the center, right? This is your genetic library. It contains all of the chromosomes for that organism inside. I should caveat that it contains most of the genes. We'll talk about, remember going back to the endosymbiotic theory, you've got a few genes hanging out in chloroplasts, you've got a few genes hanging out in mitochondria as well, but the vast majority of your genetic information is here in your nucleus. While that cell is not dividing, it's all spread out, kind of reminds me of spaghetti in a bowl, and we talk about it as chromatin. Once we get to cell division, remember it all condenses into those X structures you might be used to seeing in terms of chromosomes. Also in the center here sits your nucleolus, right? And this is where we manufacture our RNA or ribosomal RNA. And so that's where we're going to build components to your ribosome structure that will then exit your nucleus and get put together out in your cytoplasm to actually make those ribosomes so that we have those protein factories. You also have a nuclear membrane, so that's going to separate that genetic information from the cytoplasm. That nuclear membrane also connects directly to your rough ER, so they are sort of continuous with each other. So you will see the rough ER right up against the ribosomes in your cells. You also have nuclear pores, right? So these are the ways to allow material to pass in and out, mainly things like those are ribosomal RNA molecules or your messenger RNA to get out to your cytoplasm. And then it's also studded with a bunch of ribosomes as well, just like we see with your rough ER. Speaking of the ribosomes, right, these are the protein factories. So they do contain two subunits, which are built in that nucleolus. You get your large subunit here and your small subunit here. They kind of remind me of hamburger buns, right? And we are literally going to sandwich messenger RNA in between those two subunits when we get to talking about translation, right? The use of genetic information to build proteins. These guys can be bound to your nuclear membrane, they can be bound to your rough ER, or they can be free floating in your cytoplasm. And generally, depending on where they're located in the cell, drives what ultimately happens to the proteins that they produce. If they're free floating ribosomes, usually those proteins then will hang out in the cell and be used there. If they are bound to the rough ER, for example, then the proteins that they produce usually get packaged up and shipped outside the cell. When we get to looking at that large subunit in some more detail later this year, we'll also talk about these three binding sites, your P, your A, and your E sites. And so these are recognition and binding sites for going through translation. Where do your tRNAs read the genetic code? Where do they start building your peptides? And then where do we get rid of those tRNAs once they've dropped off their amino acids? That's where those three sites come in. So we'll talk a little bit more about those later this year as well.